Welcome, ladies and gentlemen. My name is Arthur Hahn, and I'm the Vice President of Marketing for Predictive Biotech. Before we begin today's presentation, I wanted to give you a little background about our company. We are a subsidiary of Predictive Technology Group, which develops molecular diagnostic tests for hard to detect and harder to diagnose diseases. Predictive Biotech was started with its primary focus on the research and development of companion therapeutics for these diagnostic tests. And it was during our research we were introduced to human cell and tissue products that were derived from birthing tissues. As we took a deeper look into these tissues, it became clear that there was a potential for clinical application. This realization was supported by researchers from around the world and from the scientific data that was being accumulated for regenerative medicine. From there, we poured our resources in developing our line of human cell and tissue products that we have today. So where does better begin? For us at Predictive Biotech, better begins with science. We want to share our research and our findings to help everyone be better informed. This presentation will go into depth of why we chose the tissues we use in our products and the steps we take to ensure safety, quality, and consistency of the human cell and tissue products that we process. Now, I'd like to introduce you all to the man behind the microscope, Dr. Doug Schmid. Dr. Schmid has over 26 years of experience in cell biology and research. He graduated with a PhD from the University of Utah in human physiology, with his specialty being in cellular biology. He spent 15 years at the University of Utah performing renal and vascular research, and it was his work with umbilical cord blood in 2007 that sparked Dr. Schmid's interest to review other various birthing tissues, which ultimately resulted in his fascination and obsession with the unique properties of Wharton's jelly. He is an avid hiker who has literally climbed every summit in Utah, and a man who I consider a goat on the trail and in the lab. Dr. Schmid, how you doing?
Doing well, Arthur. Thank you. We wanted to talk today a little bit about regenerative medicine and how it's important for the providers who are looking into using regenerative medicine in their practices to understand some of the differences that can be found between the different tissue sources. We're going to focus today on Wharton's jelly, and we're going to look specifically at the key components of this tissue, products that are derived from that tissue, and take a deep look into what these products contain. Well, why Wharton's jelly? Back in 2016, when we were starting Predictive Biotech, we looked at a lot of different tissue sources. We had an underlying need, which was to find a product that could repair, replace, or provide structural support for damaged tissue. There were several solutions available to us at the time. On one end of the spectrum, we had pharmaceuticals. On the other end of the spectrum, we had surgery. But right in the middle, bridging the gap between these two technologies was regenerative medicine and human cell and tissue products. The reason we chose umbilical cords specifically were that they had a very high number of MSCs within the tissue. They had high concentrations of growth factors and cytokines. And the viscosity of Wharton's jelly was also very interesting to us as well. And we'll touch on that as we go through this a little bit more. The umbilical cord itself is an organ. And it's important to understand what an organ is as we talk about these types of things. So let's start with a definition. So an organ is a group of different tissues working together to perform a common function. A tissue is a group of similar cells and cell products that work together to perform a specific function. So if we look at the histology of the umbilical cord, you can see several different types of tissue that stand out right away. First and foremost is the amniotic epithelium. So it's an epithelial tissue that lines the outside of the umbilical cord itself. On the inside, we have muscle tissue that is part of the umbilical blood vessels that run throughout the cord, the two arteries and the vein. In between, there are two different types of connective tissue. On the outside of the cord, there's a dense connective tissue. It's a tough outer covering that helps provide structure and strength to the outside of the cord. The other type of connective tissue found within the cord is Wharton's jelly. It's more of a loosely organized connective tissue as well. And we're going to spend some time talking about Wharton's jelly right now. So if we take a closer look at Wharton's jelly, there are a couple of things that stand out. First and foremost, we have the mesenchymal stromal cells that are found within the Wharton's jelly, and they're interspersed within the Wharton's jelly itself. There are also fibers that are found within this Wharton's jelly too. There are collagen fibers, elastin, and fibers that run throughout this tissue. And if you look at this picture, you can see that these fibers are very loosely organized and form more of a webbing than a structural support. And this is important because Wharton's jelly itself, which you can see here in the picture as a clear areas in the staining, The Wharton's jelly itself has the consistency of an egg white. And so if left unchecked, it will run to one end of the cord, and basically it has no function if it runs to one end of the cord. So these fibers, although loosely organized, maintain the Wharton's jelly in place. The Wharton's jelly itself is made up of glycosaminoglycans, things like hyaluronic acid, some of the sulfates that you see listed here as well. And one of the really interesting things about this material is how it's organized. It's basically got a brush-like structure with hyaluronic acid forming a core of the brush, and then the sulfates forming the bristly edges. This structure, this three-dimensional structure that's found within Wharton's jelly, resists crushing and kinking. It provides a site for cells to adhere and bind in it, and allows them to transmit signals between each other. The cellular distribution within the umbilical cord is also very interesting. And one of the reasons for that is because the Wharton's jelly itself does not have a dedicated blood supply. So if you look in this picture, you can't see other blood vessels running out to feed the Wharton's jelly. And so what you find is that the cells themselves, the mesenchymal stromal cells that are found within that Wharton's jelly, generally tend to be in very high numbers, very close to the umbilical blood vessels. They have to rely on the diffusion of nutrients across that vessel wall in order for them to get the nutrients that they need to grow and survive. So the majority of the cells are found in what we call the peravascular region. You can see that as you get closer to that amniotic epithelium and on the top of the umbilical cord itself, that the cells become much more sparse. And this becomes important because when we're looking at early versions of our Wharton's jelly allografts, one of the first things that we would do is that we would dissect the cord and we would remove these umbilical vessels before we process the tissue. And one of the things we quickly saw was that when we did that, we were getting cell counts that were much lower than we expected. We're also finding that our growth factor and cytokine levels were very low as well. And we immediately came to the conclusion that removing those vessels and from the tissue also removed the Wharton's jelly and the cells that were found within that tissue as well. So when we realized that the removal of these blood vessels was detrimental to our expected outcomes, the product that we were hoping to make, we immediately changed our processes and started to work around that by leaving the umbilical vessels in the tissue while we were processing. And so one of the things we'd like to do now is take you through the day in the life of an umbilical cord here at Predictive Biotech. First and foremost, this whole process begins with a pregnant mother. The mother has to decide to donate the tissue. We cannot purchase the tissue from mother. Mother has to donate it to us. The mother and the father are screened. Before they donate the tissue, they have to be screened. They fill out a very lengthy document that looks at health histories. It looks at travel histories, behavioral histories, even genetic histories, to determine if there is a risk in using that type of tissue. The mother also has to undergo a blood test. The blood is collected within 7 days of the birth, and then it's shipped off to a third-party laboratory where it's tested for bloodborne pathogens such as HIV and hepatitis. Now, at the time of birth, this is where it really begins. So, baby is born, and the umbilical cord and the birth tissue is collected, and it's collected by someone who's specially trained to do so. They collect that tissue, they package it in a very specific way, and they ship it to us here at Predictive Biotech. Once it arrives, our quality control team takes over, and they check that tissue in, make sure that all of the documents match up, that the donor numbers are correct, and that it's what we expected to get. They also look at the packaging itself. Is it damaged in any way? Is it packaged correctly? Is it the correct temperature when it arrives? And if it manages to pass those criteria, then the tissue moves on into the processing laboratory. If it doesn't pass those criteria, the tissue has to be discarded. So one thing that I'd like to do now too is take a look at the processing and kind of pull back the curtain a little bit and let you see. We'll give you more of an in-depth look at how we process here at Predictive Biotech. So the tissue will come in, and the laboratory will check the tissue in as well. So once it comes in, they will go through some quality control checks. They'll look for the color of the cord. They'll look at any type of blood clotting that is present in the cord. They'll look at how long it is, things along those lines, just to make sure that it meets our specifications for processing. If it meets those specifications, then the tissue is rinsed in an antibacterial solution. Now, using an antibacterial solution to wash the cords, we have to ensure that the antibacterial material does not end up in the final product. And so we have a third-party testing group that looks at that as well and makes sure that our rinsing procedures and our cleaning procedures are sufficient to remove all of that, all of those types of contaminants. Well, once the cord tissue is in this antibacterial solution, the technicians will begin to clean the tissue. Now, as I mentioned, we do not remove the umbilical vessels. We leave them in. And so we have to remove any type of blood or blood residue from the tissue. And that's basically done by squeezing the cord and removing those clots and whatever blood is present in the tissue. Now, if you remember your blood physiology, there are billions of red cells per milliliter of blood. So although we can clean it very well, it's almost impossible to remove all of the red cells from this final product. But we do our best to get as much out of it as possible, but we can't always get it all. And so that's one of the caveats to processing and leaving those blood vessels in. But as we mentioned earlier, we can maintain the cell numbers. We can maintain the growth factors and cytokines that are found in that perivascular region if we leave that umbilical cord vessel in the tissue. Once it's processed, we need to get rid of the material that we don't want. If you remember the histology, we're looking specifically at isolating the Wharton's jelly. We don't want the tough outer covering of the cord. We don't want the amniotic epithelium. We don't want the muscle tissue that's found within this umbilical cord. And so the processing, the tissue then goes through a filtration system to remove those contaminants. It will remove the muscle tissue. It will remove the amniotic epithelium tissue, and that dense connective tissue. What we're left with is a pure Wharton's jelly product that contains the growth factors, cytokines, and the cells from that tissue. Once the tissue is processed and filtered, it is mixed with a cryopreservant and then placed into cryostorage. This cryostorage also acts as a quarantine period for the allografts while we collect all of the relevant information for the donor, for each donor. That'll include all of the medical histories that we've collected. It will include the bloodborne pathogen testing that was done. It will also look at the testing that is done for microbial screening. We want to make sure that the products don't have any type of contaminants found within them. At this time, we're also looking at particulate content in the product, because during any type of processing, there is the possibility that particulates could be generated from the processing mechanisms. We want to make sure that those are not there. We also look at bacterial endotoxin levels that are found within these products as well. So while the product is in quarantine, we're collecting all of that information and making sure that it's complete for each donor. Once we get all of that information collected, it goes to the medical director, who will look at the donor information and make sure that that donor is a suitable donor for this tissue. If it passes his screening, it then moves on to our quality assurance department, who looks at all of the associated paperwork and all the other testing that goes along with this product to make sure that it's passed, that it's met our specifications. Only if it passes the medical director and the quality assurance review can the product be released to be used in a clinical setting. So one of the things that we wanted to discuss here is a marketing piece that's been released by another group. And in this marketing piece, one of the things that they look at is the amount of human hemoglobin that's found within several different products. As you can see here on the graph, there are 3 products that are compared in terms of human hemoglobin content. Now, one of the things that we wanted to point out on this graph is the label on the y-axis on the left. Now, this label is in nanograms per milliliter and 60,000 nanograms per milliliter. If you look at the external umbilical cord or external UC product on the right-hand side, follow the top of that bar across, it is at about 60,000 nanograms per mil. Now, that sounds like a really high number, but in looking at these data, that number is generated from a standard curve that is part of the assay that was used to measure the hemoglobin. But if we talk about hemoglobin in a clinical setting, the numbers that are used there are a little bit different, and the units that are used there are very different. So nanograms per milliliter were used in this assay. In a clinical setting, we generally see these things in grams per deciliter. And so what we've done here on the right-hand side is we've taken 60,000 nanograms per milliliter, and we've converted that into, on the top line, we've converted that into milligrams per deciliter. The reason I did that is because I didn't want to divide by billions. I thought a million was a little more manageable. So 60,000 nanograms per milliliter is 6 milligrams per deciliter. Now, if we go down to the second line, you see that 6 milligrams per deciliter, if we change that to grams per deciliter, is 0.006 grams per deciliter. So that 60,000 nanograms per milliliter looks incredibly impressive on that graph, but if we look at it in a clinical setting, it's 0.006 grams per deciliter. Now, the comparison here that we would make would be PRP injections. Now, PRP injections, one of the criteria for a PRP injection is that hemoglobin content is less than 10 grams per deciliter. So you can see that if we compare it to something that's used quite often, 0.006 grams per deciliter is about 1,000-fold less than what's actually a specification for a PRP injection. Now, all of this tissue is processed in our ISO 13485 certified laboratory. And I specifically point out ISO 13485 because this looks specifically at our quality systems. When ISO comes to certify our facility, they look specifically at whether or not we have the appropriate systems in place, the appropriate controls in place, and whether or not we're actually following those. So having that certification demonstrates that our quality system is up to snuff. We are compliant with 21 CFR 1271 regulations and also compliant with current good manufacturing processes and current good tissue processes. These techniques that we've developed in the laboratory help us to limit variations that can occur from one allograft to the next. We've shipped over 140,000 different units, and these safety records and standards continue to lead the market. One of the other things that we found really interesting about Wharton's jelly was its viscosity. As I mentioned earlier, it has the consistency of an egg white, and we thought this was really interesting. And so we partnered with Thermofisher to take a look at the viscosity of Wharton's jelly itself. Thermofisher utilized a system called the Hockey Mars 60 rheometer system, and basically what happens is it has a spinning plate, and as we move that plate faster, we apply a shear force to this fluid or to this material, and we want to see how it reacts, if it reacts in the same way as other bodily fluids. So on the next slide, there are a couple of different expected results. Now, most fluids, such as water, if you apply a shear force to it, the thickness or the viscosity of the product or of the fluid does not change. And so that's called a Newtonian fluid. In biological systems, however, things like synovial fluid, they are an example of shear thinning materials. So as you apply a shear force to it, the material will actually become thinner, allowing it to flow better throughout the joint. And that's an example of what's called a non-Newtonian fluid. So let's take a look at the data. On the left-hand side, we have Wharton's jelly. This is our polysite product that we'll talk a little bit more about in just a minute. But you can see from this graph that as you apply a shear force to Wharton's jelly, that the thickness decreases. So on the x-axis on the bottom, that's our shear force. We're increasing it as you go from left to right. And the viscosity is highest on the top on the left on the y-axis. And as you apply that shear force, the viscosity thins. Now, the reason this is important is because if we look at synovial fluid on the right-hand side in this graph, this graph is actually taken from a master's thesis that was published a couple of years ago, where the researcher took synovial fluid from several different donors and then did the same type of experiment. You can see that as you increase the shear rate on synovial fluid, the viscosity also starts to decrease. Now, it was kind of a long and lengthy discussion about non-Newtonian fluids, but the take-home message here is that Wharton's jelly acts in a homologous manner to synovial fluid. As you increase the shear force to it, it thins just like synovial fluid does. And so it acts very much like synovial fluid in a joint. Now let's take a look at a couple of products that we've developed here. So the first and foremost, we're going to talk about WJ Flow. WJ Flow is a Wharton's jelly allograft that is cryopreserved. So in this product specifically, we're looking to maintain the viability of the cellular aspect of this tissue. We use flow cytometry to look at cell counts, viability, and also to identify the cells. And in doing so, we can look at things like mesenchymal stromal cells, endothelial cells, epithelial cells, and even white blood cells to see if there's any type of blood contamination in the product. We use a nuclear stain and a mitochondrial stain to look at the vitality of these cells to make sure that they're live and functioning. And in the laboratory as well, we also have the ability to look at growth factors and cytokines and to quantify the amount of exosomes. Now, in looking at the types of cells that are found within the Wharton's jelly, we've developed a flow cytometry panel. And that's what this graph is showing you right here. So on the left-hand side, we have a marker called CD146. Now, CD146 is an extracellular marker. It's found on the surface of cells, and it identifies cells that come from the perivascular region. And so these cells are specifically called pericytes. So almost 95% of the cells that we have in this product in WJ Flow are pericytes. Now, one of the things I need to mention here as well is that these are cells that are being tested post-thaw. So these cells have gone through the entire cryopreservation process and then been thawed out, and then we've done this assay. Now, CD90, the next bar on the graph, is a marker for mesenchymal stromal cells. As you can see here, about 87% of the cells also express CD90. Now, as we look down this graph, there are a couple of other markers here that you're probably familiar with. CD45 is a white blood cell marker. CD31 is an endothelial cell marker, and CD326 is an epithelial cell marker. So if you look at the amounts of these markers that are found within the cells, that are found on the cells within this product, you can see that CD45 is found on 42% of the cells, CD31 on 61%, and CD326 on about 28%. So one thing that is important to remember here is that these markers are not absolutely specific. So you can find CD90 on a lot of other cells, not just on a mesenchymal stromal cell. You can find CD45 on many different cells, not just on white cells. And recently, there's been kind of an explosion in the literature, different scientific groups looking at these markers on cells. What they've found is that in umbilical cord tissue, and looking specifically at umbilical cord mesenchymal stromal cells, or pericytes for that matter, you can find CD146 and CD45 on the same cell. You can find CD90 and CD326 on the same cell. And so this panel, which was developed with ISCT standards in mind, ISCT is the International Society for Cellular Therapies, they presented a paper in 2006 that looked at specific markers for mesenchymal stromal cells. And so in comparing these data, it's a little bit confusing for a native cell. The ISCT standards were developed for a cultured cell, not a native cell that's directly isolated from the tissue. So these numbers are a little bit confusing, but they're expected based on what we see in literature reports. But if we take those cells and we put them into culture, as we see on this panel here, something different happens. So on the right-hand side, I've put up the ISCT standards that are used in this paper that was published in 2006. There are CD11, CD34, 19, and 45, which are basically white blood cell markers. There's an HLA-DR, which is a marker of self. It identifies cells that come from a certain person. CD73, 105, and CD90 are the positive mesenchymal stromal cell markers that they have published. Now, if you look at the graph at the table in the middle, we looked at CD11b, 34, 19, and 45 along with HLA-DR all in one group. And as we put these into culture, those markers disappear. They go away. CD73, 105, and 29, which is a different MSC marker, but also a very important one, 91% expression, and CD90 at about 93% expression. Now, these are primary cultures. These are cells that are freshly isolated from the cord and put directly onto a dish. Now, CD31, again, an endothelial cell marker, is about 0.7%. CD146 is a little bit lower than what we saw before. It's about 54%. And CD326 is about 0.3%. So when the cells come out of the umbilical cord itself, a native cell, the expression of these markers is very different than what we see when we put them into culture. Once we put them into culture, they begin to act exactly like ISCT says they should act. But as a native cell, they're very different than a cultured cell. One of the things we wanted to do as we were looking at developing Wharton's jelly flow is that we wanted to make sure that we could maintain the viability of the cells throughout the cryopreservation process and do that with less variability. Now, one of our previous products, a product that is now a subject of an IND, Coresight, was our marker, our test marker as we looked at this variability in the freezing and thawing of the product. What you can see on the left is that Coresight pre-freeze in the blue and post-thaw in the orange has quite a bit of variability when you look at the average number of cells that are present in those allografts. And that variability is represented by the black bars that are found on top of those columns. The wider the black bar, the longer the black bar is, the more variability there is between samples. So in Coresight, what we saw was that there was about 30% variability in the product, pre-thaw and pre-freeze and post-thaw. What this meant was that we had to set certain minimal standards to ensure that we got a consistent number of cells for a clinician to use once they thawed the product. With WJ Flow, we've worked hard on the cryopreservation substance or the cryopreservation solution that we use. And what we can see is that the variability, the width of those bars, the length of those bars is much lower. So where Coresight was about 30% variability, WJ Flow is now only about 10% variance. And so with this new product, with making WJ Flow, we've been able to reduce the variability within the product itself so that we can deliver a more consistent allograft. One of the other things that we wanted to do in the development of WJ Flow is not only to maintain the cellular aspect of the tissue, but also to maintain or better preserve the growth factor and cytokine content found within the product itself. So in this graph, we've taken and we're comparing that previous version, Coresight, to WJ Flow, and looking at whether or not we were able to accomplish this. So in this graph, we're using an in-house assay, looking at growth factors and cytokines. We use a MagPix instrument, which uses a Luminex assay in order to measure these growth factors and cytokines. You can see that in most cases here, such as TIMP1, LAP, which is a surrogate for transforming growth factor beta, cystatin, galectin-3, and soluble ICAM, that in the WJ product, we're able to actually increase the amount of these growth factors and cytokines that are found in the product. Now, in other aspects, things like MMP9, macrophage colony stimulating factor, and IL-37, Coresight still has a little bit more than the WJ Flow product. But for the most part, if we look at all of these products, overall there is an increase in the amount of growth factors and cytokines found within this product. Now, we've also looked at this in terms of a much larger sampling using a third-party assay available from Ray Biotech, which has also confirmed these findings as well. So we've been able to better maintain the cellular content. We've made it less variable, and we've found more growth factors and cytokines in that product. Let's take a look at the other one. Polycyte is another product that we make developed from the Wharton's jelly itself. Now, the main difference between Wharton's jelly, WJ Flow, and Polycyte is that Polycyte is not cryopreserved. It is an acellular product, and so there are no living cells found within this product at all. Because I don't add any type of cryopreservation media to it, there are higher levels of growth factors and cytokines. there are higher numbers of exosomes, naturally occurring exosomes per milliliter of this product. And there are higher levels of the extracellular matrix material, things like hyaluronic acids, collagens, fibrins, and elastins. One of the ways that we've looked at this are, again, by activity assays, or by growth factor and cytokine assays available both in-house and from Ray Biotech. But measuring the growth factors and cytokines is just one way of looking at these, whether or not these are present. We also need to know whether or not these growth factors and cytokines are active within the product as well. And so we've developed an activity assay. On the right-hand side, you can see some pictures that are taken from on our tissue culture camera. Basically, what we've done here is we've taken human keratinocytes, which are a type of skin cell. We've grown them on a tissue culture dish. Now, we'll let those cells grow until they cover the entire surface of the dish and these cells themselves, they don't like to stack on top of each other. So they form a really nice, what we call a monolayer, just one cell thick, and it covers the entire plate. So once they cover the entire plate, we take a pipette tip and we scratch across the surface of that monolayer to remove some cells. Basically, what we do when we make this scratch is we make a defect in that monolayer. Cells in a monolayer recognize their neighbors. They know that there's not any room for them to grow and divide, and they can feel that they have another cell next to them, and so they stop growing. When they recognize that there is now space where this defect has been made, the cells will start to grow in and take up that space. And you can see that. You look at the top row, the top three pictures on the control side. Now, these cells just get a normal growth medium that the cells normally grow in. So at day 0 is when we made the scratch. At day 2, you can start to see along the edges that the cells have started to grow into that space. They're starting to take up that space. At day 4, more cells have grown in and they've started to take on more of the regular morphology of a keratinocyte, more of that square kind of cobblestone look. Now, on the bottom, what we've done is we've taken that regular growth media and we've supplemented it with polycyte. We've mixed it with polycyte. So at day 0, you can see that clearly defined defect in the cellular monolayer. At day 2, however, the cells have completely grown in and covered up that space. At day 4, that space is again still covered, but the cells are starting to become more of that mature phenotype. So what we can see here, and again, I need to stress that this is an in vitro assay. This is not something that we've seen in humans or any type of in vivo situation, but this is just an assay for us to look at whether or not these growth factors are active within the cells. And from this assay, we can demonstrate that they are. On this picture, in this graph, we've basically taken and given you all of the data that we obtained from Ray Biotech. Now, again, there are 440 different growth factors and cytokines here, along with the levels of these proteins found within these allografts. So I'll save you a lot of trouble. First and foremost, the thing to look at here is when looking at the products that are derived from the Wharton's jelly that contain the most growth factors and cytokines, polycyte contains the highest levels of these growth factors and cytokines. WJ Flow contains the next highest amount. And the reason, again, for that is we have to add a cryopreservative to it, which will kind of dilute the Wharton's jelly or displace some of the Wharton's jelly. And so those levels are just a little bit lower than what we see in polycyte. Okay, so speaking of cryopreservation, now cryopreservation is a means to maintain cells indefinitely by slowing down their cellular processes. And by slowing them down at liquid nitrogen temperatures, we're taking these processes to almost a complete stop. And when I talk about cellular processes, I'm talking about protein synthesis, things like that, just the things that a cell normally does. So we're going to take those processes, we're going to stop them. It's also a means to minimize any type of genetic drift within the cells or any type of morphological changes that can happen to the cells while they're being frozen. Now, cryopreservation of a cell means that I want to disrupt the ice crystals formation within the solution. And by doing that, I'm going to keep the cell from bursting or breaking or perhaps being cut by those ice crystals. I also need to employ rate-controlled freezing. I need to freeze at a specific rate so that the cells don't shrivel up and die or or burst, as you can see on the right-hand side. So if I follow these procedures, if I add a cryopreservant to disrupt the ice crystal formation, and in this case we use dimethyl sulfoxide or DMSO, and we use rate-controlled freezing, we can then take the cells, put them into a liquid nitrogen storage container where the temperature is maintained at lower than negative 136 degrees Celsius. We can maintain those cells for many, many years. Now, saying that, we also recognize that there are some cultured cells, things like cell lines, and some tissues, things like amnion, other types of tissues, that can survive being stored at negative 80 degrees, so a much warmer temperature than what we're prescribing here. However, it's important to remember that at minus 80, even though it's very cold, these cells and tissues still have functioning cellular processes. They're moving very slowly, but they're still moving. And so in this case, you can see the cells begin to age. Now, Predictive Biotech, within our laboratory, we've experimentally demonstrated that if we take native cells from the umbilical cord tissue and add the cryopreservant, rate control freeze them, and then put them minus 80 degrees, they only survive for about 10 days at that temperature. Therefore, we've moved to a liquid nitrogen storage to help maintain that cellular viability as well. So hopefully the take-home message that we've provided you here is that it's really not just one thing. We've spent a lot of time talking about cryopreservation and things like that and the cellular aspect of these tissues, but that's just one part of these allografts. These allografts contain growth factors and cytokines. They contain exosomes, and it's this whole mix that gives the efficacy to these products. It's not just one piece.
Well, thank you, Dr. Schmidt, for that amazing presentation, and thank you all for joining us here today. If you have any questions, please visit us at our website at predictivebiotech.com or send us an email. All the links will be available in the description below. Have a great day, and remember, better begins with science, and better begins here.